This is a video which runs through our specifications in biology, and this is exactly what you need to know. And it's a fantastic tool because it enables you to see both areas where you might have some gaps, but also because of the very prescribed nature of mark schemes, they have to tell you exactly what they want you to know. And so these form very, very concise and accurate notes. And I think if you can know these well, they're an excellent base from which to build. So starting off straight into it, we need to know about cell biology and we need to know about cell structure. We need to know about prokaryotes and eukaryotes. Prokaryotes, pro means before, are those cells which are bacteria which don't have a nucleus. They have kind of like a little bit of a circular piece of DNA. Whereas eukaryotes are the plant cells which have a nucleus and animal cells which have a nucleus. So we need to know the difference between prokaryotes and eukaryotes. Then you see here we need to know about the terms centi, as in centimetre, millimetre, micro and nano. Just so you know, centi is 10 to the minus 2 metres, milli is 10 to the minus 3, a thousandth. Micro is 10 to the minus 6 and nano is 10 to the minus 9. All these would be meters. And if you have any issues with those, please contact me. Okay, so we talked about cells, eukaryotic, prokaryotic, and their sizes and scales. Okay, now in terms of moving forward, we then go into more details about cells, and you can see here that we have animals and plant cells. We have the list of the organelles that we need to know. And this is really helpful because it tells you what is in each of the cells, which organelles. So for example, in animal cells, we have the 5 organelles that we need to know about. And then we see that there's an extra 3 organelles for plant cells. It talks about the required practical activity which is here, which is using a light microscope to observe and label cells, which you remember that we did. We then move on to cell specialization. And cell specialization is the cells which have special structures relating to their functions. And they list exactly the ones you need to know. Sperm, nerve, muscle in animal cells, root hair cells, xylem and phloem in plant cells. So, once again, a really useful tool talking of exactly what we need to know about cells. We move on and we talk about cell differentiation. Cell differentiation is about stem cells and the fact that early on there are just a few cells after fertilization, and as these cells divide and divide, they slowly begin to turn on and off DNA within those cells, and they turn into loads of different cells. And so that might be brain cells, liver cells, lung cells, kidney cells. And so the same DNA can differentiate into different cells. You can see here, which is an important point, animal cells can only differentiate early on in their development. So we only had stem cells, which were these cells that could differentiate very early on in our life. Whereas plants, you'll see here, can actually retain the ability all the way through their lives, which is why you can actually take little cuttings from a plant and make whole new plants. We then move on to microscopy, and we need to know about the differences between electron and light microscopy, and once again use those prefixes because it's going to be relevant. What they might do is they might, in a question, give you a picture of a cell, and that would be an animal cell because it has no fixed shape and it's got a nucleus, and they might ask you to measure this on the page. And if they said that that was about, say, let's say it measured at 5 centimetres on the page, they then might say that it is magnified by 250 times. And they may ask you to find out the real image. And the way we do this, I'm sorry, but this equation here is not come out on the page. But what we would do is we would say, well, we've got the image size that we've measured. So we've got the real image there, and we've got the actual size here, and we've got the magnification. And we would take that 5 centimeters, which is the real image size that we see, and we would divide it by the magnification in order to get the real size. So 5 centimeters divided by 250 will give us the actual size. And we would then need to manipulate this triangle. If they gave us another thing, they might give us, for example, the size of a cell is 50 micrometers. It's magnified by 250. How big would it be on the page? And so we just use this triangle in order to work those out. And it's a bit like the speed, distance, time triangle, but just for magnification. We do need to know the differences between light and electron microscopes as well. And you can see what they're saying there in the specs. We then come to cell division. Chromosomes, which are found in the nucleus, made of DNA. And then we come to mitosis and the cell cycle. Remembering that in mitosis we have a cell which will have the DNA in, and it will form 2 identical daughter cells. And we don't just say identical, we say genetically identical. And it is used in growth and repair. And one set of chromosomes will be copied into 2, and then the cell will divide. And it is not to be confused with excuse me, the other process, meiosis, which makes gametes. So mitosis is for growth and repair, and it's where the chromosomes and the DNA copies, and then the cells divide. So we move on, zooming through these now, and what I'd like you to do is to look at these in more detail, of course. Now, we talked about cell differentiation, and there's a little more detail on that with stem cells, which are those early cells which are able to turn into any future cells. And you can see here that they tell you exactly what you need to know about stem cells. This is an example where these specifications are almost like notes. So, for example, it says that here, if we look at this, we can see treatment with stem cells may be able to help conditions such as diabetes and paralysis. In an exam question, you might be asked specifically, what are the advantages of using stem cells in medicine? And there they've given you the answer. Okay, and so it's worth using these as excellent notes. Okay, another one here, which would translate straight into a question: the use of stem cells has potential risks such as transfer of viral infection, and some people have ethical or religious objections to stem cells. And so once again, there could be a question saying, what are the issues with stem cells or the problems about stem cells? And there is the answer within the specs, which is excellent. And so we move on from stem cells and we move on to transport in cells. And this is the diffusion, osmosis, and active transport. And you need to know these really well and know when they're relevant. So we have a definition of diffusion here. It's the movement of particles from an area of high concentration to low concentration. And what, once again, these specs are very, very useful for is giving you examples of what you need to know. And so you can see here we see that they talk about carbon dioxide, and they talk about oxygen, and they talk about urea being a waste product from cells, and therefore you can see which substances they want you to talk about in diffusion. Oxygen and carbon dioxide are particularly important for the circulation system, which we come on to later. It then talks about the factors which affect diffusion. Difference in concentrations. So a very high concentration, a very low concentration would create a very steep concentration gradient, but a high concentration going to a less low concentration would be a less steep graph, so it wouldn't happen as quickly. Temperature, more kinetic energy for the particles, randomly colliding and therefore spreading out the particles, and the surface area also increases the diffusion. This is a really important one and often overlooked, and that is the surface area to volume ratio of an organism. Remember, something like a single-celled organism is able to get its nutrients fast enough through simple diffusion from the outside because the diffusion distance is so short and the surface area to volume ratio of the tiny cell is very large. But if you were to have an animal like us, we can't get through diffusion alone things like oxygen to the cells in our body, so we need a transport system. Okay, and so this talks about diffusion and those considerations. We then move on to osmosis, which is the movement of water from an area of high water concentration to low water concentration down a concentration gradient, but it has to go through a semi-permeable membrane. Really important to know that. And you'll remember that we did an experiment on potatoes, and this is another required practical here where you can see that we need to be able to talk about the effects of osmosis. So you may remember we took little sections of potato and then we put them into different concentrations of sugar solution, and the question is, would osmosis cause the water to enter the potato or for water to leave? It would enter if there was a more dilute solution around the potato, because that would be a high water concentration going into a local water concentration in the potato, but it would leave the potato if there was a more dilute solution in the potato and it was more concentrated sugar outside. Okay, and once again we need to remember the way that we did those practicals. The last of the transports is active transport, and this is, instead of going down a concentration gradient, we go uphill. And just like you pushing a ball or a bike uphill, the key about active transport, it's a movement of particles against a concentration gradient from an area of low concentration to high concentration, so the opposite of diffusion and osmosis, but it requires energy. Okay, and so we have our transport in cells. We then moved on to organisation. Very, very quickly and very easily, we have the order of the way that organisms are arranged, and we can see here that we just need to know about cells, so we could actually put the cells here, going on to tissues, organs, organ system, and lastly organisms. And you've just got to learn these definitions that are here and be able to identify them. So you might have, for example, the lungs are an organ, but they are within the respiratory organ system of lungs and heart and blood vessels. So, pretty simple one there. We just got to know those definitions though of cells, tissues, organs and organ systems. Now we come on to enzymes then, and enzymes are within the digestive system. And with enzymes, we need to know about the lock and key hypothesis. We need to know about the different enzymes that exist in the digestive system and what affects enzyme rate. We need to know about temperature and pH changing the shape of active sites if they are not the optimum. And that's why amylase in the mouth, for example, which operates normally at a pH of 7, can't work in the stomach where there's a pH of 2. And as I say these things, I'm talking about different enzymes, amylase, protease, pepsin, lipase, carbohydrases. You need to know all those. You need to know where they're found in the body, in the digestive system, and you need to know what pHs they need. Then we come on to the heart and blood vessels and the contents of blood. So the circulatory system is there designed to deliver oxygen and glucose to respiring cells. The heart, the lungs, the blood vessels, the different types of blood vessels are important here. And this is, once again, a really good way of checking that you know everything. So this tells you about which are the structures in the heart you need to know. And then the 3 types of blood vessels that have different features depending upon their function. Blood itself consists of plasma, red blood cells, and white blood cells with platelets in, and they all have a function that we should know about. And lastly are the problems in terms of our circulatory system, and this is the coronary heart disease, which is caused when there is a blockage to the coronary arteries. So if you have a heart, for example, which I've just crudely drawn there, it's got 4 chambers, running over the heart, which I'll do in a slightly different colour, running over the heart, which supply the heart itself, are the coronary arteries, and they deliver oxygen to the heart tissues itself. And if we were in coronary heart disease to get some kind of a blockage as a result of a build-up of cholesterol or fatty deposits, then you can imagine that that artery is less able to carry blood. And if less blood gets through, there's less oxygen, less glucose, and it means that the cells of the heart may die. And if you have cells in the heart dying, it can't pump, and that means you don't get oxygen and glucose to the brain, and you just completely die. We then also talk about, in this area, stents and valves and transplants, which might help with issues. So that is a run through of the specifications that are going to be included in your mock. I hope you found it helpful. You can just Google the AQA trilogy specs to get this document. I believe you should work from it. I believe you should use it often. I think you should highlight those areas I said that you're not sure of. But it's a really, really excellent tool. And if you learn all of these things and you are able to look at the videos that I've uploaded, and look at my beads for all of these different areas, then you'll be in really, really good shape for the mock. Good luck.